Chapter 10. This is for muscle types and how muscles work, and we'll start at the cellular level. So, as a kind of intro, we have seen this similar slide before in the tissue chapter. There are three different types of muscle tissue. Skeletal muscle being what is most commonly studied, and it does go along with the skeletal muscles that you are learning in lab. Skeletal muscle is the one that is attached to the bones of the skeleton, which is why it's so named. It is considered voluntary, which means that we can consciously control and send signals from our control center, the brain and spinal cord, to these muscles so that they move. And you can kind of see a few characteristics here, which we'll get more deeply into for those cell types when we get to them in this chapter. So A right here is skeletal muscle, looking at what those cell types look like as a cartoon, and then also what they look like as a histological slide under the microscope. Cardiac muscle, we'll get to later on in this chapter, makes up the most of the wall of the heart. Okay, so that's the myocardium, which is the main portion of the heart, made of cardiac muscle cells. This type of tissue is involuntary, so it does move, and we know that our heart is moving all the time, but this is under what we would call non-conscious or subconscious control. And again, the main action that it is doing is the pumping action for the heart to move blood throughout the body. The last type of muscle that we'll discuss today is smooth muscle. This is found on the walls of internal organs, as well as blood vessels, parts of the eye, other areas. The example we give here is the digestive tract that you can see in this picture. And again, smooth muscle, similar to cardiac muscle, the cells work differently, but they are nonetheless involuntary, so under non-conscious control from the control centers of our brain and spinal cord. So a few functions of skeletal muscle, and we're going to focus on skeletal muscle for the majority of this chapter until we get to a few short things about cardiac muscle and smooth muscle at the end. For skeletal muscle, obviously, since these are the muscles attached to the skeleton, they help us to move the body. So when we are trying to move those muscles, we move bones, we make facial expressions, it allows us to speak, to breathe, to swallow. And although I will clarify this breathing and swallowing, those mechanisms are made of both smooth muscle and skeletal muscle. So the skeletal muscle components we can actually move ourselves. So part of swallowing is us being able to consciously swallow, at least for the first part, and same for breathing with the diaphragm made of skeletal muscle. But there are smooth muscles throughout that system too, allowing us to complete these actions. It's kind of a second function is to maintain posture, right? We have muscles kind of surrounding joints and helping us maintain body position. We have muscles for protection and support. So aside from bones that are also protecting internal organs, we have muscles around those bones, which are an additional layer of protection. Um, some of what we do with our muscles is for moving things through the tract. So we have elimination of materials. There are certain sphincters, okay, that are kind of openings. They open and close. They're circular in shape, and we can kind of control those sphincters for releasing material from the body. In one case would be excretion of feces. And then another really big function for skeletal muscle is the production of heat. Our muscles generate a lot of heat when we use them, and that is part of what helps us maintain our body temperature. So if your muscles are not moving, your body temperature is going to get lower. Or if you, are doing normal muscle movements, producing body heat, but you might be in a cold environment, moving those muscles more will increase your body temperature. A few other things that we will get into, which will be a bit more complicated in this chapter, are the characteristics of skeletal muscle. And when we talk about things like excitability and conductivity, we're going to be talking about electrical impulses that are going through these cells and changing in what we call resting membrane potentials for each of these cell types. Excitability, which we'll get into, is the ability to respond to a stimulus by changing electrical membrane potential. All cells kind of interact with other cells at the membrane level first, right, at the cell membrane. And so when the cell membrane's electrical potential changes, we have charges running through them, it affects what certain pumps or channels open, it affects what the cell takes in or what it gives out. So we'll talk about that. Conductivity involves sending an electrical change down the length of the cell membrane. Okay, so some cell types are more conductive than others. They have certain things there that help them to conduct that electrical signal to the rest of the cell. Specifically for muscles, they have contractility, right? So they're able to contract and distend. So we will see that for muscle movement. A lot of times when we view those cells that have those striations, it kind of looks like an accordion, which we could move in and out. So moving closer together, those fibers would be contracting, and then moving away would be relaxing. So we'll talk about that. Some muscles have a higher degree of extensibility, so the ability to be stretched, and then some elasticity. So if we lengthen or shorten those muscles, they're able to go back to their original shape. So we're going to look first kind of at the, gross anatomy of skeletal muscle. Each skeletal muscle is considered an organ for the musculoskeletal system, and they all work together to perform a specific function depending on what, muscle group we're looking at. There is associated other things with these muscles as part of this system, like connective tissue, blood vessels, and nerves. It's important to note, we will get into this distinction, but we're going to be talking a lot about muscle fibers. And what I wanted to note there is the word fiber for the musculoskeletal system is the same as saying a muscle cell. So instead of calling them all muscle cells, we'll call them muscle fibers. You can kind of see in this picture here, okay, we have kind of set up little sections of this muscle, and you can kind of tell this is a thigh muscle because we've got a femur here. And at that smallest division, we have the muscle fiber. Okay, so that's our skeletal muscle cell. We put a bunch of those together, okay, into a bundle, and that bundle is called a fascicle. And then in a muscle itself, we have a group of fascicles that are all put together, and you can see that there's varying membranes surrounding them, which we'll go into. And we have, they're nicely packed together with these membranes, these connective tissue layers to form our actual muscle group. So for those membranes or those coverings of connective tissue, I have kind of two renditions. So this is a blown up picture of what we just looked at on the previous slide, and then this is kind of a nicer cartoon picture to kind of show that relationship. So first, for these coverings, we had already learned something kind of similar when we were in the bone or skeletal chapter, where we have these coverings on the inside and outside the bone. And if we remember, those are called periosteum and endosteum, okay? Not too different when we get to muscle. Of course, we're going to have kind of a different root word here, this mice, right? For epimysium, perimysium, endomysium, that my, that's the root word, and that means muscle. Epimysium is going to surround the entire muscle. So if we were talking about, we have the cell goes into bundles called a fascicle, and then we have a bunch of fascicles making up the muscle. All those fascicles are brought together and surrounded by epimysium, epi meaning on the total outside, and then that mice for muscle. For each fascicle, okay, that we create, bunch of fascicles in the muscle, they're surrounded by their own connective tissue layer called perimysium. Perimysium, peri meaning next to, mysium again meaning muscle. Okay, so we've got that bundle surrounded by perimysium. These are surrounding our fascicles. And then lastly, we have the cell itself, our muscle fiber, and it is surrounded by its own connective tissue layer called endomysium, endo meaning the most internal. You can kind of think of this like maybe if you were collecting firewood and you were putting it into bundles, okay? Surrounding each individual stick or log would be a endomysium, right? Surrounding each individual thing. You put them together in nice little bundles or packets, and that is surrounded by perimysium. And then if you put all those packets together, maybe to take them to a different location, they would all be surrounded by and kept together by this epimysium on the outside. So we're going to keep going into some of the other gross anatomy or additional structures that are part of this. Okay, so this is kind of the general formation that we saw in this previous slide for how we form muscles. But these muscles, as you can even see here in this picture, have to be attached to bone through other avenues other than just these muscle coverings, one of which is going to be a tendon. So we'll take a look at that on the next slide. So a tendon is a cord-like structure of dense regular connective tissue. We know what dense regular connective tissue is. It what forms our tendons and ligaments. It's just this nice organized arrangement of collagen fibers. So tendons are going to connect muscle to bone. And remember, we learned the other term ligament, which connects bones to bones, and it's part of that joint capsule. There are a few other words that we can use for connective tissue parts for attachment of muscles. One is aponeurosis, and the other one is fascia. Aponeurosis is thin, flattened sheets of dense, irregular connective tissue. Okay, if we remember that type of tissue. Dense irregular is more randomly organized. It has a little bit more flexibility and stretch to it, right? Than tendons made of that dense regular tissue, which are just those nice organized collagen fibers that are tightly packed together. Dense irregular for our aponeuroses is a little bit less tightly packed, so it allows some stretching, some movement. You might have already noticed an aponeuroses when you were studying your muscles for lab. There is an aponeuroses between the frontalis and occipitalis muscles in the head. We learned the muscle occipitofrontalis that is connected by an aponeurosis. And most of the time, when you do look at these models or pictures of tendons, aponeurosis, or fascia, it's going to be white tissue on any kind of model or picture. And so it might not surprise you that aponeurosis is dense irregular connective tissue that is connecting other muscles to each other. So we're not necessarily using an aponeurosis to connect muscle to bone, but we are connecting muscles to each other across maybe a wider area. And then we have fascia. So we have what we call deep fascia and superficial fascia. We learned these terms already in the early chapters, chapter 1 even. Deep meaning this is going to be kind of innermost, very deep into the body. Superficial meaning surface level. So we'll see this kind of on the surface for certain types of muscles. So it might not surprise you that superficial fascia separates muscles from the skin, right? That's more towards the surface. And superficial fascia is mostly made of areolar and adipose connective tissue. Deep fascia, okay, so this is deeper in the body, not towards the surface, is going to be lying kind of right next to epimysium. So remember, epimysium is what covers that entire muscle group. It covers all of those fascicles. And so this is sitting on top, this deep fascia made of dense irregular connective tissue, very similar to the aponeuroses. And what it's going to do is separate individual muscles from each other. So how, even if we go back to our leg example, here, I'm not sure what muscle this is. This is on the front of the leg, so this might be maybe one of those thigh muscles, okay? Maybe it's sartorius, okay? That seat belt across the thigh. How we separate sartorius from all of our not our glute, excuse me, all of our quad muscles or from some of our other muscles like gracilis, adductor longus, all of those different ones is with this deep fascia. So this is dense irregular connective tissue separating individual muscles from each other in a muscle area. For additional structures, we'll just mention blood vessels and nerves. We're not going to really get too much into what specific ones they are until we get to the nervous system chapter. But skeletal muscle is vascularized, meaning there are blood vessels there, okay? The reason for this is because since we are constantly moving these muscles, we definitely need a good nutrient supply and oxygen supply so we can produce ATP. And then we have production of different wastes that need to be taken away from those cells. Muscle is also innervated. This is how we do that voluntary control with skeletal muscle, right? We have to have nerves connected in order to send signals. And largely, what we would call those nerves are somatic motor neurons. Neurons, again, being the name for our nerve cells. Somatic means the body, it also means voluntary, okay? And motor meaning things that are going to move. Those muscles send signals for movement. So let's kind of get into it a little bit deeper into the muscle cell itself. Okay. This is not the same picture as before. You can see in this tiny picture up here, we have that whole muscle, we have a fascicle, and then we have our muscle fiber. This is the muscle fiber blown up. And within the muscle fiber, we have our own smaller structures, okay, that kind of look like they are individual cells, but they are all just part of this one muscle fiber, one muscle cell. Okay, so I have our skeletal muscle fiber. If you noticed in some of the previous pictures, you might notice here as well, they are multinucleated, so multiple nuclei here. They have kind of a difference in naming for some of the structures on these cells, different from our composite cell that we learned in chapter 4. So instead of us saying we have a cell membrane on our fiber, we have a sarcolemma. That is the name for the cell membrane, sarcolemma. And it contains, and this will become important later, voltage sensitive calcium channels. Okay, so little holes in the membrane that allow calcium to go in and out of the cell. Okay, voltage sensitive calcium channels. Voltage sensitive meaning that we need to change the electrical potential of the cell in order to activate the channels opening or closing. So the cell membrane for a muscle fiber is sarcolemma. Next, if we have the cytoplasm inside the cell, remember cytoplasm is the cytosol with all of the organelles, then we have a different name for that for the skeletal muscle fiber. It's called sarcoplasm. Okay, sarcoplasm. And then within here, we will see, you've probably seen on your lab that we had like a kind of muscle cell present. There is a ton of other organelles like mitochondria and things like that. But all of these individual structures that you see within the muscle fiber are called myofibrils. Myofibrils, and there are hundreds of thousands per cell. Few additional structures. Okay, so myofibril, we will get into these thin and thick and thin filaments, which are part of their contractile units. One other additional structure that we kind of see overlaying inside the cell is what we call a sarcoplasmic reticulum. Very similar to what we might think of for the composite cell, like a smooth endoplasmic reticulum. Sarcoplasmic reticulum is going to play kind of a different function. It's going to store and release calcium when our muscles need it. So we'll get more into that function a little later, but that's another structure inside this cell. So getting to these myofibrils, if we pull out an individual one, which is what you see here in this picture, there are different filaments present, which are going to glide across each other, either getting close together for a contraction of that muscle cell or getting farther away from each other when we relax that muscle cell. These filaments, which we will kind of designate what all these different parts are, but these filaments that move across each other are called thin actin filaments and thick myosin filaments. And again, thick and thin, those words tell us kind of a description of the fibers. Actin filaments look thinner, myosin filaments look thicker, and then actin and myosin are the names of the proteins that make up these filaments. So for our filaments, okay, and we have a picture of each of them. This top one here is the thick filament made of exclusively myosin. And you can see that these myosin filaments also have these little heads that come off. That will be really, really important because it's going to be their attachment point to the other fiber, which is going to be the thin actin filament. So this is our thick filament made primarily of myosin proteins, okay? And then they have these little kind of what we call myosin heads coming off. Thin filament here, a bit more complicated as you can see. So the main part of this filament is made of actin, okay? So we can kind of see this string of pearls here. There's two strings of pearls kind of wrapped around each other. Those are actin. That's protein for actin. And then we have two other proteins, which we call regulatory proteins, which are sitting on the surface. The green that's kind of wrapping around, okay, is called tropomyosin. And then the other one, which are these kind of blue looking ones sitting on top here, are called troponin. Okay, lots of T words here. So we have actin making up most of the thin filament, but we have two regulatory proteins sitting on the surface, which are called tropomyosin and troponin. These are going to be really, really important because these are kind of providing the binding sites so that we can bind the myosin heads to this filament when we are doing a muscle contraction. So if we look at that kind of organization even further, okay, so we're not looking at this up close version of these fibers, okay, but you can kind of see these fibers here, right? We can see the thick myosin filaments, which are this kind of Barney purple here, okay, and then you have the thin actin ones, which are that kind of periwinkle purple, I guess you would say, kind of surrounding here. So the thick ones kind of towards the center of this fiber, and then we have those thinner actin ones. These are kind of placed together in little square sections, okay, if you can kind of see this whole section here, um, where we have kind of naming of different zones as well, which we'll get into. That whole little section here where we have overlap of myosin filaments and actin filaments is called a sarcomere. Sarcomere is the functional unit of a skeletal muscle fiber because it contains both the actin and the myosin filaments, which are going to be doing the muscle contraction. So we call that a sarcomere. Um, the delineation between them, delineation just meaning the beginning and the end of each sarcomere, are designated by these Z discs. So Z disc right here, Z disc right here, or you could call it a Z line as well, up to you. That's going to separate each individual sarcomere that we have present. And we have, again, hundreds, if not thousands of sarcomeres in each kind of myofibril, right, which is one of the main structures inside our muscle fiber. So let's take a look a little bit deeper into the sarcomere. So now we're actually going to be able to see the structure of the actin and myosin and that overlap. So again, we can see the myosin thick filaments here. You can see their little heads coming off. And then you can see surrounded by these thin actin filaments, which, look a little bit more complicated because they have those regulatory proteins present. And then if we want to just have one sarcomere, it goes from Z disc to Z disc, right? Okay, so we're just going to focus in right here between each Z disc. All right, so there is an additional separation, okay, and these are kind of arbitrary, I would say. These were named a long time ago for kind of separating out different parts of the sarcomere. Okay, so we're going to look at first at I bands and A bands. Okay, anywhere where we have just thin filament, okay, and you can see a little bit from the previous sarcomere. So just the thin actin filament is the only thing that we can see present. That is where we have what we call our I band. Okay, it appears lighter on the myofibril for the muscle cell. It's just that light area where there's only thin actin filaments. That's what we call our I bands. And when we do a muscle contraction where we are going to be moving these actin closer over the myosin heads for a muscle contraction, it will appear that the I bands get smaller during a muscle contraction because they're moving closer together and overlapping more with the thick filament. For the thick filament, you notice that there is a small part, and this is relaxed muscle right now, okay? There is a small part where it is just the thick thick myosin filament, and that small part there, as we kind of look at here, we'll have what we call an H zone and an M line, which we'll get into on the next, actually, we have it here. So H zone and M line, these are all just consistent of only that myosin thick filament. Okay, M line is just the center of the sarcomere. And then the H zone is just the area where there's myosin, just like the I band is just where there's areas of actin. And then that overlap here that we see, which should be kind of the darkest part of this sarcomere, where we have overlap of thin and thick, we would term our A band. Overlap in here, thick and thin filaments. And you can kind of see a representation of this a little bit by color in this bottom picture. So that's kind of our main parts that we see for the sarcomere. And again, these are just designations so that we know what parts we're looking at when we actually do a muscle contraction. And so the next section for this is that we are going to go into a muscle contraction. So for the contraction of a muscle fiber, there are multiple steps. I tried to separate them out for you as A through E that we'll kind of look at. And what we're mostly getting at is the shortening of these sarcomeres. So this previous picture here, we have our sarcomere and it is in a relaxed state. But as we do a muscle contraction, all of these fibers are going to get closer and closer together and they're going to kind of converge on this M line, right? We're all getting really close together towards the M line and the muscle is going to contract. Very similar if we are kind of compressing an accordion closer together, okay? And then when we relax, we move them farther away from each other. So overall, when our sarcomere is contracting, we see it shortening, okay? Because those muscle fibers are getting closer together. And we're going to follow this kind of step by step, okay? We're going to start where we need to start, which is that nervous system stimulation, so neurotransmitter junction, and I've given you the neurotransmitter that is starting the muscle contraction, which is acetylcholine. We're not going to learn too many neurotransmitters in this class that you have to memorize. I think that is something much more reserved for a neuroanatomy class. But to start with, this is probably the only one you have to memorize for this course. We're focusing on acetylcholine. Okay, acetylcholine is the neurotransmitter that starts this muscle contraction. We then move to the next step where we activate the sarcoplasmic reticulum. If you remember from a couple slides ago, that is a structure within the muscle fiber that is similar to the smooth endoplasmic reticulum, but instead of it storing lipids and processing those, it's storing calcium and either releasing it or storing it for later. Next, we go to an excitation phase where we have contraction coupling. So coupling meaning those fibers are going to come together. We have something called cross-bridge cycling, which we'll go into more, where they go across each other and do an action. And then when that whole entire muscle contraction is finished, we will go through some of the process for relaxation of that fiber again. So we'll kind of start here where we have a structure. All right, so here we have the spinal cord, okay? We have some nerve fibers, okay? And again, this is really just one neuron in this case, okay? Or we've got two here that have axons, which we'll get into a little bit later. Axons are the part of the neuron that communicate with other cells. So we can see these axons here, and they are eventually touching, okay, communicating with these muscle cells. And we can tell that these are skeletal muscle fibers because of all the multiple nuclei. They have these striations, which are those lines that are formed when we have the sarcomeres present. Everything we need to know. So the first step, okay, is that communication. And when we have the axons from a neuron, from a nerve cell, communicating with a muscle fiber, we call this whole thing a motor unit. Okay, so the motor unit becomes active where we have these motor neurons sending a signal to the muscle fibers as part of that motor unit. These motor units are established, but they're not always stimulated, right? The way that we stimulate this system is by releasing neurotransmitters. So kind of our first step, okay, is we can kind of see here. So again, we still have a motor unit here, and right where we have that connection, so this is the end of the axon, and you can see it's kind of laying right next to the muscle fiber. There's this kind of area here, And this whole area is a space we would call the neuromuscular junction. Neuro meaning the part from the nervous system that's touching, and muscular, the part from the muscular system that's touching, and the junction is just the connection between the two. These aren't actually touching each other. There is a space here, okay, which we call synaptic cleft, which we'll get a little bit into, and we'll get more deeply into for the nervous system, but they are in communication with each other. The way that they communicate is through all these very small structures you can see at the end of the axon, which are called neurotransmitters. So again, neuromuscular junction, location where the motor neuron innervates the muscle, and it has a series of parts. First one being the synaptic knob. So each of these kind of expanded ends of the axon is called a synaptic knob. So this whole area is a synaptic knob. We have a synaptic cleft, so just this space, okay? You can think of it kind of as just an opening, right? In between a small space, in between where the nerve is and where the muscle is. And then we have what we call a motor end plate. And the motor end plate is on the muscle fiber itself. And that is where on the motor end plate, it can actually receive the neurotransmitter that this synaptic knob is going to release. So this is a blown up image of this, okay? Right here we have synaptic knob, yes, okay? We have the space here, synaptic cleft, and we have the motor end plate on the muscle end, okay? Just try to follow the pictures the best you can. So our synaptic knob is part of our motor neuron. So our motor neuron is going to send an impulse, okay, to this muscle. Synaptic knob is what houses our synaptic vesicles, okay? A vesicle, if you remember, because we talked about vesicles, especially with the Golgi apparatus, when we were making proteins in our composite cell, they package those proteins into vesicles so they can move throughout the cell. So a neurotransmitter is just a protein that this cell has made and packaged into a synaptic vesicle. So this synaptic vesicle is basically just a vehicle housing or holding onto all this neurotransmitter inside. So these vesicles are going to make their way to the end of the synaptic knob. They're going to fuse with the membrane and they're going to release their neurotransmitter. Neurotransmitters are just chemical messengers released by motor neurons. Okay, so in our chemical messenger, we know, okay, because it's the only one we have to memorize, is ACH, which is just the abbreviation for acetylcholine. Okay, so acetylcholine is being released from those synaptic vesicles. And the only way that this acetylcholine is actually released is from calcium interacting. Okay, so we see these voltage-gated calcium channels. When calcium is flooding into the cell, that's going to trigger the synaptic vesicle to fuse with the membrane and release acetylcholine. Acetylcholine, you can see, is kind of floating around here with sodium in the synaptic cleft, and it's making its way to some of these receptors, okay, present on the motor end plate, okay? And you can just see that this receptor is just called an ACH receptor. So an acetylcholine receptor that's present on the muscle fiber to take in that acetylcholine. And again, that's going to be on the motor end plate, the part of the muscle. So a few things are going on, and this is where it gets a little more complicated for that electrical potential. It's not too bad, okay, but a little more complicated. So kind of the first step, and again, always refer back to this picture. This is a good picture kind of explaining this through. So we see we have these voltage-gated calcium channels on the synaptic knob of the neuron, okay? So calcium enters the synaptic knob, okay? And that nerve, so how that happens, okay, we have a nerve signal that travels down the axon. And again, the axon is just the part of the neuron that communicates. It's like an extending arm that communicates with other cells. And so when it becomes stimulated and a nerve signal, it's an electrical signal, travels down the axon and eventually makes its way to the synaptic knob. And when that electrical potential changes, it opens these voltage-gated calcium channels. Calcium is going to diffuse or flood into the synaptic knob, and it's going to bind to proteins on the surface of those synaptic vesicles. Again, those vesicles are just vehicles holding neurotransmitter. Since calcium is binding to those synaptic vesicles, it's going to cause those vesicles to merge with the cell membrane, and we're going to release our neurotransmitter, ACH. And that release is just called exocytosis. This is bringing back some of the terms we learned about movement across the cell, right? We have diffusion of calcium coming into the cell, and then we have exocytosis of that ACH once those vesicles merge with the membrane of the synaptic knob. And again, those ACH, those neurotransmitters, are going to be released, thousands of them, right, into the synaptic cleft. Okay. So we see all of that ACH is diffusing across the synaptic cleft, and it's looking for those ACH receptors, okay, on the motor end plate. So it's going to go to the motor end plate, and ACH, when it binds to its receptor, it's going to cause electrical changes in the membrane of the muscle fiber. And what we would call that is exciting the fiber, right? ACH is an excitatory neurotransmitter. When it binds to the motor end plate on those receptors, it's going to excite the muscle to want to contract. And again, I'm just using the abbreviation ACH. You could learn, well, you should learn that it's acetylcholine, but on a test, if you wrote ACH as your answer versus acetylcholine, that would be perfectly acceptable. All right, so we're going to take just a very, very short view into what is happening with the electrical potential. Okay. We're going to get much more deeply into this for the nervous system, but I wanted to touch on this first in this chapter before we get too deep in the weeds when we get to the nervous system. Okay, so I'm going to just walk you through this just briefly, okay, and we'll get more deep into it before. This is a graph of membrane potential. So this is the electrical potential of the cell membrane, okay? And we know that we have electrical potentials built up in cells because when we were learning about all those subatomic particles, you have protons, neutrons, and electrons, all of them giving off their own charge, okay? And largely, when we have a group of those molecules all come together to form our cell membrane, largely for all cells, the cell membrane potential is net negative. Okay, so our typical membrane potential that we would say for most cells, okay, and we'll say this one for specifically the muscle cell, is negative 90. Okay, so net negative. That's our what we would call resting membrane potential. So when the cell is not stimulated or doing anything, it is at this negative 90. Okay, and that's normal. The cell's just not doing anything. The cell needs to reach a certain threshold, okay, for it to be excited to start doing activity. And the threshold for our skeletal muscle fiber is negative 65. So it needs to become a little bit more positive, okay, from this resting membrane potential of negative 90. How is that going to happen? Okay, how that happens is we have the opening and closing of different chemically gated ion channels. Chemically gated ion channels. And we care about these ions because they all have their own charge. Okay, the specific ions that we focus on, especially in the nervous system, are going to be sodium, Na+, potassium, K+, and calcium, Ca+. Okay, or Ca plus 2 in theory. Okay, so once we try to reach our threshold, and this would be through some electrical activity, okay, these chemically gated ion channels, they end up opening, and our cell is flooded with sodium, okay, so Na+. Sodium is going to come in, okay, sodium is coming into the cell, and since sodium has a positive charge, it's going to change the charge of the resting membrane potential to be slightly more positive, right? We're still in the negatives, but we're slightly more positive here at that negative 65, okay? All because sodium is flooding into the cell and it has a positive charge. The more sodium that floods into this cell, we start to have this kind of steep climb up here. And that process where sodium is coming into the cell, okay, and it now turns into voltage gated because we are increasing our voltage, okay? That climb is called depolarization, where the cell is becoming more and more electrically positive because of all that sodium coming in. And we eventually reach kind of a climax here for depolarization, which is for the muscle cells about positive 30. Okay. And this means once we've reached this climax that we are going to do our action, whatever it is. In the case of the muscle cell, we're going to do a contraction. Okay. So this is called depolarization. Okay. And during this depolarization phase, okay, the action potential travels along the sarcolemma. Okay, remember sarcolemma is just our cell membrane and causes the release of calcium from the sarcoplasmic reticulum. So the sarcoplasmic reticulum in the muscle fiber cell is just storing calcium. And when it becomes activated, okay, because we are creating this action potential here through depolarization, those calcium ions are stimulated and they release from sarcoplasmic reticulum. And those calcium ions, we're going to talk about in a minute, are going to bind to something, one of our muscle filaments, so that we can start our muscle contraction. At the end of our muscle contraction, we do go through a period where we go downhill called repolarization. This is where we have sodium channels close and we open up potassium channels, and potassium floods out of the cell, making us go back down to this negative point. We'll talk about that a little bit later. So again, right now we're just talking about creating an action potential. We want to do the action of muscle contraction, and that means we need to reach a threshold, and then all of a sudden we are doing this depolarization. We are creating an action potential so that we can do our muscle contraction. This, at this point, is just involving sodium. Sodium's coming into the cell, making it more positive. Once that action potential travels to the sarcolemma, it's going to interact with sarcoplasmic reticulum to release calcium. Calcium is then going to go to our sarcomere and bind to a specific protein. So our next stage, calcium is going to bind to troponin. Okay, if you remember troponin when I was describing those different filaments, remember we have a thick myosin filament and a thin actin filament. The thin actin filament had two regulatory proteins on top of it. One was troponin and the other one was tropomyosin. Troponin is where the calcium is going to bind. And troponin, when calcium binds, is going to cause a change, a movement in that complex. Typically, I'll kind of state this, troponin needs to bind to calcium in order for it to move. And tropomyosin is kind of just a standby protein. What it's doing while the cell is relaxed is just blocking the binding of the myosin heads to the actin filament. But when calcium binds to troponin, troponin pushes tropomyosin out of the way. We have a change in how the fiber is oriented, and it leaves up these open binding sites for myosin heads to bind to the actin filament, forming a structure we call a cross bridge. So once we move out of the way, right? Troponin gets bound by calcium and it pushes tropomyosin out of the way. We now have these open active sites for the myosin head to bind to. Once they bind together, we do something called cross bridge cycling. So the binding of a myosin head to the actin filament is called a cross bridge. And then we do this 4 repeating step cycle for as long as calcium is present, pushing troponin and tropomyosin out of the way so that we can bind. First is the cross bridge formation. So the myosin head attaches to the exposed binding site on the actin filament. Next, we do something called a power stroke. The myosin head pulls the thin filament towards the center of the sarcomere, which is that M line, okay? And during that process, we actually hydrolyze ADP and our inorganic phosphate here. So it was ATP, but we've kind of broken up that bond. Then we have a release of the myosin head. ATP is going to bind again, causing it to release. And then we reset where we have ATP coming in, hydrolyzing, and then causing that connection to form. And we do the power stroke all over again. A little bit complicated. I'm going to show you guys a video, which will definitely help with that. This is a picture rendition, although I find the video helps a lot better for kind of visualizing what's going on. Um, And let me see, I definitely posted the video in here. Yeah, so this video, we're not going to watch it together, but it will go through each of those steps for cross bridge cycling. It's very, very helpful. So pull up that slide for how the cross bridge goes, follow the video. It'll help you understand it a lot better than maybe I can explain to you, just especially if you're very visual with that. I'm going to go back real quick. So once we do this cross bridge, and you can kind of see in this picture here, the sarcomere is shortened, showing that it's contracting. We do eventually, because our muscles do not stay contracted unless we are in rigor mortis, okay, which we'll talk about briefly. Um, our muscles, for a person who is alive, they do go back into a relaxation state. What happens for muscle relaxation, those events? We terminate the nerve signal, which means that ACH is not going to be released from the neuron anymore. There is a protein that is an enzyme here called acetylcholinesterase. We know it's an enzyme because it has that ASE, and then it tells us its substrate in the beginning of the word. So acetylcholinesterase is an enzyme that breaks down acetylcholine. So any acetylcholine that's left in the synaptic cleft is going to get broken down, hydrolyzed by acetylcholinesterase. We have closing of our acetylcholine receptors. So on the motor end plate, we're not going to be accepting any extra acetylcholine. There's no action potential generated. We have calcium channels closing on the sarcoplasmic reticulum, and a lot of the excess calcium is going to return to the sarcoplasmic reticulum. Troponin goes back to its original shape, meaning tropomyosin is now blocking the binding site for the myosin heads, and the muscle returns to its original position. Okay, that's part of our muscle relaxation. All right, so we'll skip the video. So, in the case, if your muscles stay partially contracted for a long period of time, and every one of us has probably seen roadkill before. Okay, I chose that as our kind of picture for rigor mortis, or you've heard of rigor mortis described in forensic file kind of cases. So skeletal muscle, when an individual passes away, can remain partially contracted for several hours after death. As you can kind of see with this deer here, instead of all of its legs kind of just laying off to the side, they are in this kind of upright position, as if you were to stand the deer, they could stand because those muscles are contracted. This typically can last for up to 72 hours, okay? And eventually, those contractions will break down. So rigor mortis is not something that just stays forever. The cause of this is because calcium is leaking into the fiber and it's forming those cross bridges. Remember, calcium binds to troponin. Troponin pushes tropomyosin out of the way, and then we have attachment, right, of our muscle fibers to each other. Okay. And so this can happen where we are forming these cross bridges, which we just call cross bridge coupling, because of that calcium leak. There's no ATP going to come in and bind, which causes the separation of the myosin head from the actin filament. And so these muscles just stay in a constant contraction. Okay, eventually these cells will disintegrate. We won't have calcium kind of perpetuating this process anymore, and eventually the muscles will become flaccid. But rigor mortis is something that can set in within the first few hours of an individual's death, and it can last for a fairly long time, for several days. So for this kind of second part here, and I'm going to post a second video about this, we are going to talk a little bit about how we make all this ATP or energy for our cells in order to continuously do this process of forming those cross-bridge cyclings and doing muscle contractions. So stay tuned for an additional video after this, just explaining this last process.